Welcome back, class. Today we are going to demonstrate the practical on polymerase chain reaction, otherwise known as PCR. So make sure that you've read this prac thoroughly before going any further with this video series. All right, let's start by taking a look at the reagents. So the reagents required to set up a PCR reaction includes this RedTac, ready mix DNA polymerase, which comes from Thermus aquaticus, or rather the enzyme, the polymerase enzyme comes from Thermus aquaticus, and this is a premix that contains not only the enzyme but also DNTPs, that is your 4 nucleotides, A, C, G and T, some loading dye, and magnesium chloride, which is an important cofactor in the PCR reaction. We also need some PCR grade water, primers diluted to a working concentration, DNA to be amplified, that's our genomic DNA that we extracted from wheat, dedicated pipettes, a thermal cycler, which we'll show you later on, sterile filter pipette tips, PCR tubes, and that's it. So let's have a look at that. So we'll start over here with our ready mix, RedTac PCR reaction mix with magnesium chloride added. Here's our PCR grade water. Our genomic DNA that we extracted, ready for amplification. We're going to use 2 sets of primers. So here we have a forward and reverse primer called TA ARF, where the TA stands for Triticum aestivum, which is a scientific name for wheat. Here we have another set of primers. These ones are called TA CDC. That's the forward and that's the reverse primer. All right, we're also going to use different types of tubes. These are only 0.2 mil flat cap PCR tubes, in contrast to the 1.5 mil microcentrifuge tubes that we used for extracting genomic DNA. All right, let's look at what they look like. Bob's already set these up for us. 2 tubes have been set up. One for one set of primers and one for another set of primers in the reaction mix. We have our dedicated auto pipettes here and some sterile tips.